Okay, so I was asked to introduce myself. I think my name is Heinz Moser. I'm a synthetic chemist by training and basically joined the pharmaceutical industry more than 3 decades ago. Time is flying. And so I've done medicinal chemistry in multiple areas, multiple disease areas. Probably a little over 15 years ago, I kind of got approached or moved into anti-infectives and found it very fascinating, but also extremely difficult. And I think my talk is going to go a little bit about trying to share some of my experience. And I'll promise you I won't have all the answers, but I'll at least try to explain why we thought it's not a trivial area for some pretty specific reasons. So let's see here. So just Andrea, first thanks to a great introduction. It saves me time to talk about some of these particular items, and I'm going to focus really on antibiotics in particular. The one thing, just as a reminder here, and Andrea already mentioned this is a multi-parameter optimization process, drug discovery and lead optimization in particular. And while property space is important, it certainly isn't the only solution. So please keep that in mind. And just to add on that, I will focus on the property space. There are many other things that matter. We'll see differences in bacterial species. There is some literature out there that will talk to that. They are active transport mechanism. I'll leave it to other sessions to argue if that's a good or a bad thing. Target inhibition requirement. I will show you an example, a specific example on that, because then that can vary substantially. And there are, of course, other parameters. It is a challenging field, but it's also fascinating in that particular part. So what I want to do is giving you a little introduction. Most people here know that it's difficult to identify antibiotics, so that's certainly not news to you. But then I'll talk about the property space and what I think matters to that in this context. And I'll give you, at the end of the talk, some thoughts for the future, where I think this field could move and where actually some promising areas are. So you all, or most of you, have seen this paper. It's kind of a nice example of negative results, so to speak. SmithKline-Beecham did a large campaign to really find novel antibiotics, and David Payne and co-authors eventually published that in 2007. By this time, I already knew over dinners and glasses of wine from literally most large pharmaceutical companies that went through exactly the same experience. And it's also kind of a reminder that we shouldn't only publish positive data, but actually the field can learn a lot about failure as well, and preventing that and focus and move on. So nevertheless, we, to the best of my knowledge, don't have a drug that resulted from that campaign, and this is largely talking about gram-positive only. It's not even touching the gram-negative field. So it is difficult. There are certain challenges in antibiotics, and I think it's fair to say that most of the field struggled really. You can get the biochemical hit, but converting that hit to a wholesale active drug that basically will penetrate the bacterium and not cause other effects, often what people have found during that optimization campaign. You can kill bacteria in many different ways, and as you're going to go through the lead optimization, you'll actually often deviate from the mode of action and will eventually start killing the bug in different ways. There is a resistance generation. Lynn Silver has mentioned that many, many times, not only she, but I think we're all familiar with that if you go after a single target, it's going to be an issue. You're basically going to invite the bacterium to prevent it and basically become resistant against it. The other thing that we often forget, I've worked in other disease areas, including oncology, inflammation, respiratory diseases. You often talk about very little drug exposure. I'm sure Many of you know what Lipitor is, and you know that the doses vary between 10 and 80 milligrams, 80 being the highest. Here we often talk about grams, multiple grams of doses. Huge exposures we typically require, and there are good reasons for that, but they offer their own problems. Andrea talked about solubility. It's a different beast if you have a 12 gram that you have to provide to a human being a day as opposed to a few milligrams. And that requires decent safety and good PK behavior. And then I think we'll certainly talk later on about the broken economic model, which is an all different story in itself. So why is it that antibiotics are so different from many of the other disease areas? And I think a lot of it lies in the architecture, and let me kind of focus in my talk more on the gram-negative side, which I think we all kind of shifted from the 80s maybe, where Staph aureus was the major focus of a lot of research campaigns, to gram-negatives. The cell wall, the architecture of the cell wall is different. So in addition, so I'm kind of simplifying here, but I think it serves a purpose. The inner membrane of a bacterium is not too dissimilar to a mammalian cell. And I think we do understand, we can model, we do understand permeability parameters on that inner membrane reasonably well. Then you have the peptidoglycan, which I look at it like a rebar around the cell to kind of keep its shape as the outside environment changes, and that is really not the barrier for permeability. But then on top of it, on gram-negatives, you have that outer membrane, which is basically has its own lipid A, which sits on the outer side, so it's an asymmetric cellular membrane, and that particular lipid membrane is almost impermeable to molecules. And that is really what kind of causes the problem, one of the biggest problem I think in this area. How do you deal with permeability where you have two membranes that require opposite parameters, and then on top of it, it's like the icing on the cake, you have efflux that really tries to prevent molecules from entering the cell. And so here a picture I showed before, just a little bit a more molecular representation of that membrane. So permeability, it's not really permeable on the outer membrane, so how do molecules get in? Nikaido published a long time ago and basically talked about the porin proteins embedded, which is an entry route, one of the major entry routes for molecules. Now think about it, this is like physics in certain ways. It's a cylindric hole that sits in a membrane that is impermeable, but the inside of that hole is highly polar. So it's going to be very difficult to squeeze a very lipophilic molecule because you have to dehydrate that surface area first, which energetically is almost impossible. So what this asks for is polar molecules on one side, but on the other side it's also like a molecular sieve. It kind of filters for molecular weight. Now on the flip side, you have the inner membrane, which actually beneficially prefers lipophilic molecules so they can enter. So you have two membranes that kind of ask for the opposite, and that makes it very hard to find molecules that will do both. Now on top of it, you have all the efflux pumps, which especially in Pseudomonas, as you know, there are multiple of them, and they're promiscuous. They're basically optimized over a long period of time by evolution to really efflux a whole variety of molecules. And sometimes you're amazed how diverse they can be, that one efflux pump can, for instance, efflux both fluoroquinolones and aminoglycosides, which to me is pretty amazing because those are quite different molecules in terms of their properties. So when we started when I joined the QIAGEN in 2005, and the company that was designated or basically wanted to make gram-negative compounds, and knowing basically that the industrial model to do high-throughput screening of corporate archives wasn't really that successful, we basically had to figure out, what we wanted to do and how we wanted to approach that problem. And one of the striking things that I realized is that nobody had done a serious analysis of the existing property space of antibiotics that made it into the clinic or even onto the market. And I talked to Chris Lipinski, and Chris actually confirmed that nobody ever had done it. There were some literature studies, but what they didn't do correctly, according to my understanding, was actually binning the compounds correctly. Now, I just showed you the different architecture of gram-positives and gram-negatives, so you can't throw all the antibiotics in one bin. You have to separate them. And what we basically did is, as you show here in the slide, we defined different bins. So gram-positive activity only is compounds that basically work as defined here on the slide against gram-positive organisms, Staph aureus in particular, and we basically looked either for ATCC strains, so there is no resistance that plays into that game here, or MIC50. Sometimes we could find one value, sometimes we could find the other. What we do know is that in most cases, these two values are actually within about the twofold of each other if you have statistically significant data, which means n doesn't equal 1. On the gram-negative side, and a lot of those gram-negative molecules are active on gram-negatives. E. coli here, again, as defined, actually do have activity against gram-positive organisms as well. There are a few exceptions. I mean, think about LPXE, where you shoot after a target that is only present in gram-negatives. So LPXE, a healthy LPXE inhibitor should basically be inactive against gram-positive strains. So there are exceptions. Then in addition, we wanted to compare those compounds to a drug space, and we used the comprehensive medicinal chemistry database and basically threw out all the antibiotics because we didn't want them in there, and we threw out all the other. It's actually amazing if you go through that database how many compounds are in there: nutrients, diagnostics, peptides, meaning greater than 4 amino acids, so there are antibodies in there. We wanted to do that comparison with a low molecular weight space. So this is what we've done. This is basically setting the stage for our analysis. So what I'm going to show you now is, and this is we published this in 2008, I believe. So this is now an expanded set. So we added more compounds as we learned more and basically updated that data set. So this presentation is basically cumulative fraction versus, in this particular case, molecular weight. The black line on the left represents the drug space. And as you know from the literature, somewhere in the order of 330 or 340 is your average weight, molecular weight of a small drug. If you now compare the gram-positive in red and the gram-negative in blue, they both shift to the right, higher molecular weight. What is remarkable is that almost 25% of the gram-positive drugs are actually over 1,000 molecular weight. But think about vancomycin, right? Vancomycin doesn't have to permeate the cell. It works on the peptidoglycan, which is easily accessible on the outside of a gram-positive cell, but it doesn't work against gram-negative because it has no chance to enter either probably of the membranes. On the other side, the gram-negative one, what is remarkable there, and again, I want to cite Hiroshi Nikaido. He published a long time ago that actually the pore entry will filter for a molecular weight of roughly 600 Dalton. Now look at that curve. I mean, I didn't bias the curve. It basically shows that over 95% of the drugs are below 600 molecular weight on the left-hand side. And hence confirmed. What was a little bit surprised is that the molecular weight is actually a little bit bigger than the average drug. So now let's look at the same, but look now at lipophilicity, as again Andrea introduced the terminology. So it's actually the water solubility versus fat solubility parameters. And again here, well-known drug space is somewhere between 1 and 2. This is a calculated log D, so it takes in account the ionization of the molecule, but as it's calculated, the calculation of pKa still is not perfect, so there will be some error bar, but having more than 4,000 compounds, think it's actually a pretty solid data set. Now, if you look, the gram-positive space is more polar, which maybe does come a little bit as a surprise, but it's not substantially different. However, if you look at the gram-negative space, it's more than 4 orders of magnitude more polar. And that is quite remarkable, but remember, the outer membrane and the entry gate through that outer membrane really does require polarity. And it doesn't really lag behind. If you look at the average number of 1.6 in terms of polarity of the average drug space, you hardly have any compound left that actually is more lipophilic than that. So we can present that in a different way, and here shown again, red is gram-positive only, and blue is gram-negative. And you can really see how they differentiate. Now, if you look on the left middle side, around 1200 molecular weight, you see like 3 blue dots. And I just told you that 600 molecular weight is kind of the limit, more or less. So why do you find these compounds? It's actually kind of beautiful to see. If you look in detail, those are the polymyxin and related compounds. They basically disturb the outer the membrane. And as such, they don't need to enter as a small normal molecular weight compound does. And hence, you know, you can understand why they violate that particular property. But again, it's kind of a reminder, there are always exceptions to the rules. This is not a rule carved in stone. This is just trying to learn from past compounds being successful. Now, what matters as well is, of course, as you can imagine, is where actually is your target. If your target is in the periplasm, your compound doesn't have to enter the inner membrane. However, if your target is in the cytoplasm, it has to go through both membranes and avoid efflux, obviously. And there are differences, so we can separate those two. And as you can imagine, you have, for instance, substantial differences. For instance, the polarity, so look at the second column, log D. Whereas the cytoplasm is relatively narrow, and what I've done here is I chopped off from the Gaussian distribution curve the 10 percentile left and right, and then took the middle 80% range as a range. So rather than give you specific numbers, kind of talking a little bit more in ranges here. And you can certainly see that for the cytoplasma you have more restriction in molecular weight. The log D is more narrow. You're basically very open to high level of polarity, and I will show you examples on that, which reflects as well in polar surface area, which kind of goes in parallel to the lipophilicity or polarity of molecules. And it also reflects in the H donor, so that's the proton donor number of proton donors you have in a particular molecules. And one of the parameters we haven't really talked about is free rotatable bonds, which in my view is a little bit archaic measure of flexibility, conformational flexibility. Now, Andrea mentioned that before. These parameters are interconnected, so think about it. So you have, for instance, a molecule that you have your target in the cytoplasm. Now you're optimizing your molecule. You're, let's say, on the negative side of the log D scale, so you're on the polar side. Now you increase the molecular weight and still retain your high level of polarity, you're going to get a penalty on the inner membrane. So if you're actually doing that, it ain't going to work. So you either need to let go a little bit of that polarity, or you need to lower the molecular weight. And so there is a connection between these parameters. And again, it's not rocket science. If you think about it, a lot of it makes sense. So anyway, let's move on and show you some of the examples. So what I've done here is the gray dots, the little dots, represent the comprehensive medicinal chemistry space. So you'll see actually probably the center of that, where those dots are the closest. So these are these 4,600 molecules that made it into the clinic. Probably somewhere around 2-3 log D, probably somewhere around 300, maybe a little bit higher molecular weight. You find that spot. It varies, obviously, so you see. Now, if you look at the corporate archive, and I actually did do the analysis, as Andrea did as well, on the Novartis archive. So typically what we have, and this is, I think, true for most companies, we have it shifted a little bit more to higher level of lipophilicity and to higher molecular weight. Why is that? I call it chemical convenience. It's much easier to put the phenyl group on a molecule rather than a hydroxymethylamino or whatever you want that has more functionality and is more polar. So this is a problem of our archives, and I think is at least partially responsible to what we've seen, lack of success. It's not the only cause, of course. Now let's look at that gram-negative space. That gram-negative space is fundamentally shifted to the left and maybe somewhat to lower molecular weight. Again, it does depend if your target is in the periplasma or in the cytoplasma. In that blue kind of ellipse, you would expect the cytoplasmatic compounds that work against cytoplasmatic targets on the bottom right side. And I'll show you again examples. So the first one is LPXE. Now what I've done here is we made, I think, over 1,000 compounds, so we have a pretty decent collection of compounds. What I selected here is molecules that had an MIC against Pseudomonas, ATCC, the standard strain, of 8 or less micrograms per mil, but at the same time didn't show any cytotoxicity at 100 micromolar. Cytotoxicity, as measured, we typically use 3 cell lines. Now that threw out a lot of compounds, so there is a very limited set here, and then I colored them according to the potency. The most potent one are green, and the less potent one are red. But remember, each one of them has an activity of 8 or less. And what you can see is really that they actually move out of that typical archive space quite dramatically. The most potent compounds are, again, the ones at the very bottom to the left, the green dots. Those were kind of our lead compounds. So let's look at the different target. Now, this, again, is a cytoplasma target. And here, again, same thing. Now, it's E. coli, but same rules. We basically chose these compounds. And again, you see that they kind of, it's a little bit hard to see, but the green dots are again at that bottom right side of the blue space. And so very similar. Now the optimization we did, and again here we made probably, again, around 1,000 compounds or so. So it was largely driven by the data we collected. So there wasn't necessarily only property space driven optimization. And I'll show you another slide on that particular target, which actually speaks much better to what I just said. So as a comparison, fluoroquinolones, 6 compounds, 3 of which are still clinically used, even though QRDR gets the better of them, unfortunately, David, as you certainly are aware. But you can see where they localize in this space. And 3 of them still used, 3 of them maybe less so or didn't make it. Now here, this is from our monobactam program. Now again, reflect on this. This is a periplasma target, so the target is not in the cytoplasma. The compounds do not need to get in. We know as a fact you could add as much polarity to the molecules as long as they would engage with their target, they still would actually work. And I think this picture speaks for itself. Now let's go back to a cytoplasma target. By the way, this resulted in LYS228. You've heard the story, or at least some of you heard the story. The compound currently started phase 2 clinical trials. So this is back to the second target within the cytoplasma. Now here what I show you is all the compounds. So no selection, right? And I again color them in terms of the MIC level. What to me is really striking to see. Now here, there are toxic compounds in here. Basically everything was thrown in here. But what you can see is that irrespective of other filtering systems, you really can see where the activity space populates. You have a relatively narrow range within the polarity or lipophilicity space, and you have a relatively narrow space within the molecular weight space. You can't be too big, because otherwise you're going to lose permeability. These compounds, by the way, actually have decent efflux behavior. If you get too small, you're not going to have enough target engagement. If you get too polar, you're probably not going to get through the inner membrane. If you're not polar enough, you're basically going to lose the outer membrane. So you have to optimize for all these parameters, and that, I think, really reflects a challenge. And here, one of the things I want to mention, because Andrea showed the picture about ionization, and fluoroquinolones, for me in particular, are very interesting because they're kind of chameleons, and they actually exist in multiple species. So if you look at ciprofloxacin neutral pH, you have about 5% of the protonated species, you have about 5% of the deprotonated species, you have roughly 90% of the Swither ionic species, but you also have probably about 0.2% of the neutral species. So, and they're in rapid equilibrium. And so you always have species, the positively charged one and the neutral one certainly can enter the inner membrane much more easily. And that will help. So let me show you another case. And this is basically the question, of course. What I'm trying to do here is really zoom into what really matters or what has been so hard in our area, aside from the topics I'm not going to cover, which is really permeability and efflux. Now, a lot of people have started measuring permeability, so did we. We have a lot of doubts and big question marks about the accuracy of the data. And so what we did find is another way to really measure what I think is way more meaningful than measuring the actual concentration of a compound within a bacterial cell. Those of you who are familiar with those methodologies, you have to work up cells, and so there's always an issue of redistribution. If you look like at Hergenrother's paper in detail, you will see that there is a disconnect of some of the observations and what he actually reported in terms of intracellular exposure, and we found exactly the same. So this particular target, you're probably fatty acid biosynthesis, CoAD, kind of very early on to make, for instance, acetyl or succinyl-CoA, which then kind of is being used to assemble fatty acids. I think you get access to the slides, so you can go back, and this work actually has been published this year. There are 3 papers which I give you the citations on. So you can go and actually read it up if you're interested. But I think it is an interesting story. The enzyme is well behaved. We got a lot of crystal structures. We did do a high throughput screening, and we did do fragment-based approaches. The hope was really that fragment-based approaches would allow us to really focus more on the property space. And we had like 39 co-crystal structures, so most of the work that I'm talking about, we actually know how the molecules bind. So just in a nutshell, we started from this fragment on the left side. You can see it was pretty decent already, sub 10 micromolar. We then, with structure-based design, kind of optimized it and got pretty quickly much better, to 37 nanomolars on the right-hand side. We then optimized it further, and again, those 2 papers will cover that particular work. We basically then optimized it further and got a single-digit nanomolar inhibitor. You can see decent ligand efficiency, which basically means, I think we haven't defined that particular terminology, but what it tells you is that the molecule, it's a certain level of molecular efficiency. E. coli wild type activity, and with this particular molecule, we also got a efflux pump deficient activity, but we also got wild type activity. We've never succeeded, despite making, again, probably 700 or so compounds, to get better than 32 micrograms per mil. And so what we did here, and this is really, I think, one of the key contribution we made in that particular project, which is the reason I want to show you. So what we did, instead of measuring the compound, the exposure of compound in the cell, we actually measured the enzyme inhibition within the cell. We would incubate the bacterial cell, and you have to optimize it for every target. to a certain extent, and then basically lyse the cell and determine downstream metabolites. It's called targeted metabolomics. So we can actually quantitate downstream metabolites, which are follow-up products from this particular enzyme. In this particular case, we went after acetyl-CoA, which is kind of 2 steps down or 3 steps down from CoAD. What you can see if you titrate, so look at the yellow curve. The yellow curve is the delta tol C, and I'm not going to go, we used it as a surrogate for efflux deficiency, which I know is not quite correct. But what you see here, as you titrate in the compound, you start to deplete that downstream metabolite to the extent that it's over 99% inhibited. Now, what you can do is you can actually do an EC50 within the enzyme and list it as we've done here against the concentration of your compound. Now, what is interesting, look at the MIC. The MIC is 100-fold shifted, meaning that you need probably 99% inhibition to kill the cell. And I'll show you more statistically significant data. This particular, so MIC is 0.5 on that delta tol C strain. Wild type, no MIC. However, look at the brown curve. Despite that, we already start that the molecule still works. It just doesn't work well enough to kill the cell. And then on the flip side, what we actually achieved here in that particular case is to exchange the human or the bacterial enzyme with the human enzyme, and that gives us a really nice control. The bug is very happy with the human enzyme, and it serves as a beautiful control to make sure you're still on target. So for every compound, we could actually determine, and we know that if we left target, we started killing that bug as well. So now look at this slide here. This is basically comparing the MIC of E. coli in the efflux deficient strain with basically the depletion in the same strain as measured by targeted metabolomics. So what it means theoretically, let's say if you inhibit your enzyme 50% and you kill the cell, all these points should be on the 1x curve or around that 1x line. Now what that tells you is that we have roughly a 100-fold shift plus minus. So basically we do need to inhibit that enzyme. Really, we didn't know that when we started out, but I think it's statistically a pretty significant finding. So that was helpful for us to understand. So the second one here is now we do the same. So we measure that enzyme inhibition, but now we compare the delta tol C again. We use that as a surrogate, be it right or not, against the wild type. Now here you can see that most compounds shift almost 1,000-fold. Now what that means, we did not solve the problem on efflux with that particular scaffold series, despite us trying very hard. But sometimes, you know, the enzyme, as Andrea mentioned before, you have to optimize multiple parameters. So sometimes you just lose if you do the thing that you would want to do to improve the permeability, the efflux. You're basically going to lose activity. So then finally, permeability. So this one is the biochemical IC50 on the x-axis versus the enzyme inhibition within the cell in the delta tol C strain. Now what that tells you is that for those series of scaffolds, permeability was not the issue. 2/3 of the compounds we made, statistically very significant, they shift 10-fold or less. So permeability, at least in this case, was not the issue. But what is nice about this whole approach is that this really gives you solid data you can build on. I honestly do not know how to predict efflux. I think permeability, we actually have a much better understanding, both on the outer and inner membrane, aside from active transport mechanism. So, conclusion, you know, HDS, low success rate. CoAD is not an ideal target, as I pointed out, and as I just mentioned, efflux and permeability were two different beasts. Now again, this is target specific. You might pick another target, other scaffolds, and you're going to be in a different environment. But what I do believe is creating solid data will help you to eventually solve the problem. So my last slide. Now what can we do? I think historically, phenotypic screening has been really successful, but I think we found the jewels, and we don't really find much more, that especially in the gram-negative space. What we need to do is really populate the optimized chemical property space. And I can tell you, we've done experiments with the Novartis archive, which is huge, creating certain small libraries, optimizing them for sp3 characteristics, the polarity space that I showed you, and basically in silico making libraries, with the result that not a single compound, all assembled from commercial building blocks, was present in our archive. Very interesting. So I think if we can do that kind of Dell-like, so you go for millions, high level of diversity, tens of millions of compounds, you will find active compounds. I'm pretty convinced of that, because I do know that the archives really don't cover that space well, very poorly. Certainly, I wouldn't give up on natural products. I think that's still a promising area. One of the problems that we do face in natural products is the synthetic challenges. These compounds, because nature typically doesn't optimize pK and all the parameters that you need. So you have to put in a lot of effort to make that happen. As we know, for instance, think about macrolides. And then also change the screening conditions. Well, I wrote it down here, Muller-Hinton broth does not equal human tissues, but I'll let you think about that. But I do believe we need to become more smarter than just continuing screening Muller-Hinton broth. Targeted approaches, one of the things we can do, and maybe John, you're going to address that in your next talk, is really asking the question, does your target actually allow that property space to work as a ligand or not? I think there are ways how you can do that. I talked to you about Metabolomics. I love that technology because it provides really solid data. It's not doable for everything you're going to go after, but it's actually doable against quite a number of targets. And then I still wouldn't let go on the fragment-based approach, because I think it does allow you to build, to evolve, as opposed to have a high throughput screening hit, to have to disassemble it and then rebuild it again. So with that, I want to add my acknowledgement slide. Part of that work, a lot of that part happened at the QIAGEN. So I would like to thank those people, some of which sit here, Ryan. We had many, many discussions around that topic. But also at Novartis, I had a number of colleagues, and certainly this goes beyond the people listed. So thank you.